The green algae are the large group of algae within which the Embryophytes emerged in the Charophytes. However, as the Embryophytes are traditionally excluded, they form a paraphyletic group, although the clade including both green algae and Embryophytes is monophyletic and may be referred to as the Viridiplantae and as the kingdom Plantae. The green algae include unicellular and colonial flagellates, most with 2 flagella per cell, as well as various colonial, coccoid and filamentous forms, and macroscopic seaweeds. In the Charales, the closest relatives of higher plants, full cellular differentiation of tissues occurs. There are about 8,000 species of green algae. Many species live most of their lives as single cells, while other species form coenobia, long filaments, or highly differentiated macroscopic seaweeds. A few other organisms rely on green algae to conduct photosynthesis for them. The chloroplasts and euglenids and chlorarachneophytes were acquired from ingested green algae, and in the latter retain a nuclear morph. Green algae are also found symbiotically in the ciliate paramecium, and in Hydra viridissima and in the platyhelminths. Some species of green algae, particularly of genera Charophytes of the class Trabuxphaceae and Trentperlia, can be found in symbiotic associations with fungi to form lichens. In general, the fungal species that partner in lichens cannot live on their own, while the algal species is often found living in nature without the fungus. Trentperlia is a filamentous green alga that can live independently on humid soil, rocks or tree bark or form the photosymbiont in lichens of the family Graphidiceae. Cellular structure: green algae have chloroplasts that contain chlorophyll A and chlorophyll B, giving them a bright green color, as well as the accessory pigments beta-carotene and xanthophylls, in stacked thylakoids. The cell walls of green algae usually contain cellulose and they store carbohydrate in the form of starch. All green algae have mitochondria with flat cristae. When present, paired flagella are used to move the cell. They are anchored by a cross-shaped system of microtubules and fibrous strands. Flagella are only present in the motile male gametes of Charophytes and are absent from the gametes of Pinophyta and flowering plants. Members of the Chlorophyceae class undergo closed mitosis in the most common form of cell division among the green algae, which occurs via ephycoplast. Like land plants, Charophyte green algae undergo open mitosis without centrioles. Instead, a raft of microtubules, the phragmoplast, is formed from the mitotic spindle, and cell division involves the use of this phragmoplast in the production of a cell plate. Origins: photosynthetic eukaryotes originated following a primary endosymbiotic event, where a heterotrophic eukaryotic cell engulfed a photosynthetic cyanobacterium-like prokaryote that became stably integrated and eventually evolved into a membrane-bound organelle, the plastid. This primary endosymbiosis event gave rise to 3 autotrophic clades with primary plastids: the green plants, the red algae and the glaucophytes. Evolution and classification. Green algae are often classified with their embryophyte descendants in the green plant clade Viridiplantae. Viridiplantae, together with red algae and glaucophyte algae, form the supergroup Plantae, also known as Archaplastida or Plantae sensu-lato. The Viridiplantae diverged into 2 clades. The Chlorophyta includes the early diverging prasinophyte lineages and the core Chlorophyta, which contain the majority of described species of green algae. The Streptophyta includes the Charophytes, a paraphyletic assemblage of freshwater algae from which the land plants have evolved. Below is a consensus reconstruction of green algal relationships, mainly based on molecular data. The algae of this paraphyletic group Charophyta were included before in Chlorophyta, so green algae and Chlorophyta in this definition were synonyms. Reproduction. Green algae are eukaryotic organisms that follow a reproduction cycle called alternation of generations. Reproduction varies from fusion of identical cells to fertilization of a large non-motile cell by a smaller motile one. However, these traits show some variation, most notably among the basal green algae called prasinophytes. Haploid algal cells can fuse with other haploid cells to form diploid zygotes. When filamentous algae do this, they form bridges between cells, and leave empty cell walls behind that can be easily distinguished under the light microscope. This process is called conjugation and occurs for example in Spergera. The species of Ulva are reproductively isomorphic. The diploid vegetative phase is the site of meiosis and releases haploid zoospores, which germinate and grow producing a haploid phase alternating with the vegetative phase. Chemistry. The green algae span a wide range of I13C values, with different groups having different typical ranges. Green algae contain sulfated polysaccharides like remnan sulfate in the amorphous sections of their cell walls. Some species also contain gallatannins. Physiology. The green algae, including the Charales algae, have served as model experimental organisms to understand the mechanisms of the ionic and water permeability of membranes, osmoregulation, turgor regulation, salt tolerance, cytoplasmic streaming, and the generation of action potentials. See also, brown algae, red algae, seaweed, Viridiplantae, chlorophyta. References. External links. Green algae and cyanobacteria in lichens, green algae, Monterey Bay green algae.